Researchers have used cutting-edge technology to bioprint miniature human kidneys in the lab, paving the way for new treatments for kidney failure and possibly lab-grown transplants. The study, led by the Murdoch Children's Research Institute (MCRI) and biotech company Organova, is published in the journal Nature Materials. The researchers validate the use of 3D bioprinted human mini kidneys for screening of drug toxicity from a class of drugs known to cause kidney damage in people. The research showed how 3D bioprinting of stem cells can produce large enough sheets of kidney tissue needed for transplants. Like squeezing toothpaste out of a tube, extrusion-based 3D bioprinting uses a bioink made from a stem cell paste squeezed out through a computer-guided pipette to create artificial living tissue in a dish. MCRI researchers teamed up with San Diego-based Organova Incorporation to create the mini organs. MCRI Professor Melissa Liddle, a world leader in modeling the human kidney, first began growing kidney organoids in the year 2015. But this new bioprinting method is faster, more reliable, and allows the whole process to be scaled up. 3D bioprinting could now create about 200 mini kidneys in 10 minutes without compromising quality. From larger than a grain of rice to the size of a fingernail, bioprinted mini kidneys fully resemble a regular sized kidney, including the tiny tubes and blood vessels that form the organ's filtering structures called nephrons. Professor Liddle said by using mini organs, her team hoped to screen drugs to find new treatments for kidney disease or to test if a new drug was likely to injure the kidney. Drug-induced injury to the kidney is a major side effect and difficult to predict using animal studies. Bioprinting human kidneys are a practical approach to testing for toxicity before use. In this study, the toxicity of aminoglycosides, a class of antibiotics that commonly damage the kidney, were tested. The researchers found increased death of particular types of cells in the kidneys treated with aminoglycosides. By generating stem cells from a patient with a genetic kidney disease and then growing mini kidneys from them, also paves the way for tailoring treatment plans specific to each patient, which could be extended to a range of kidney diseases. Professor Liddle said the study showed growing human tissue from stem cells also brought the promise of bioengineered kidney tissue. 3D bioprinting can generate larger amounts of kidney tissue, but with precise manipulation of biophysical properties, including cell number and conformation, improving the outcome. Currently, 1.5 million Australians are unaware they are living with early signs of kidney disease, such as decreased urine output, fluid retention, and shortness of breath. Professor Liddle said prior to this study, the possibility of using mini kidneys to generate transplantable tissue was too far away to contemplate.